So good morning, and I'm glad to be here presenting my research findings to you today. I especially thank the Psoriasis Association for giving me this opportunity to stand before you today. So my research topic is on serious infection risk associated with the biologics that are used for the treatment of psoriasis. So a small background before getting into the actual research. So it is well known that psoriasis is a problem with the immune system in which for some reason more number of immune cells get into the skin, especially certain inflammatory cytokines such as TNF-alpha, interleukin 12, 23, interleukin 17, and interleukin 23 are seen more in the psoriatic skin lesions. So conventionally we have topical medicines, ointments, creams to apply on the lesions. But if the condition is moderate to severe, we have oral medicines that could be used to manage this condition. But more recently, we have another group of drugs, as we all hear today, biologics, which are really effective when compared to oral medicines. So these biologics actually act by inhibiting or interfering with these specific cytokines that are seen more in the skin lesions, such as those are mentioned here. So when this inhibition happens, good prognosis is seen. So as you see today, more than 1 in 2 patients with moderate to severe psoriasis, they use biologics. As you see in the graph, more and more people are using biologics over the years. But with the conventional medicines, it's pretty static over the years. So the problem is, even though these biologics are effective, there is a concern regarding the serious infection risk associated with this biologics. This is especially because the biologics interfere with the cytokines that are known to be protective against certain infections, such as tuberculosis or herpes zoster, candida infections, and so on. So theoretically, it's possible that these biologics can induce serious infections. We have some evidence for older biologics, but with the newer biologics, we do not have much experience, and we do not really know the actual risk with the newer biologics. So in the past, there have been some biologics withdrawn from the market due to this serious infection risk, and therefore, it is very important for us to know if the biologics that we are using currently for this condition are really safe with respect to serious infection risk or not. So with that understanding, we aimed to estimate and compare the serious infection risk associated with all the biologics that are approved for use in the UK today. So that's what our primary aim was. And today we have around 12 biologics, and we analyzed all the 12 biologics in our research. So how we did the study, we systematically reviewed the published medical literature to see if there is any evidence for serious infection associated with these biologics. So 2 of us independently reviewed the medical literature, and if there was evidence, we collected the data, such as how many patients actually received the biologics and how many of them developed serious infection, all those related data. And with that collected data, we analyzed to estimate and compare the infection risk with every single biologic using a relatively modern method called network meta-analysis. So what's so special about this network meta-analysis is that this method is capable of estimating the infection risk with every single biologic, even if there is an absence of data with some of the drugs. So you might wonder how this Statue of Liberty is connected with the research. To be honest, it's not connected to the research, but just to explain the method. So I'm sure we are familiar with these structures here. I just want to compare the heights of these structures, and I want to know which one is the tallest and which comes second and which one is the third. So for this, I need 3 values, actually. I want to know how tall is B than A. I want to know how tall is C than A. And I also want to know how tall is C than B. If I know these 3 values, I can say this is the first, this is the tallest, and then comes this, and then comes that. But all I have is I know B is 100 meters taller than A. I also know that C is 200 meters taller than A. But I do not know how tall is C than B. So this is where network meta-analysis comes. Even if I do not know this information, network meta-analysis can come up with an estimate for this height, and it will tell you this is the tallest. This is easier for us to understand. Now, if you know these two values, I can easily say that B versus C is 100 meters. 200 minus 100, that's 100. So this is what exactly network meta-analysis does. So it gathers information from other data available, and it will come up with an estimate for the drugs where we do not have proper data. So these are our findings. We got more than 1,000 published literature after the search, but when we after reviewed the literature, we found 110 studies to be eligible for analysis, which included more than 50,000 patients. So these are the characteristics of the studies included and the patients involved in the studies. So this is a nice diagram that shows all the medicines that we compared in our research. Basically, it includes all the biologics that we currently use in the UK, and also the oral medicines that we use, and the placebo here. So the lines between the nodes indicates that there is a study or evidence comparing those two medicines. And the thickness of the lines are proportional to the number of medicines, number of studies comparing those two medicines, and the size of the nodes are proportional to the number of patients who received that particular medicine. So these are the significant findings from our research. In general, we found that the serious infection was rare in the studies. There were quite few studies which reported zero events in their results, meaning none of the patients who took the biologics developed serious infection. That's encouraging, actually. But when we put together data, we also found some differences between some of the drugs. Especially, we found one drug called risankizumab, as you see in the graph, to have low risk of serious infection than one of the older biologic called ustekinumab. So as you see in the graph, the area left to the central dotted line means that the medicines are safer, and the area to the right means that the medicines are harmful, more risk. So risankizumab, which is a newer drug, is shown to be safer than ustekinumab. And when we compared the different classes of biologics, we found one particular class that is interleukin-23 inhibitors. That's again a newer class of biologic, found to be safer than placebo. And also we found ustekinumab, just pretty old, and that was found to have high risk when compared to interleukin-23 inhibitors. So in both the cases, interleukin-23 inhibitors was found to be better than placebo and interleukin-23 inhibitors. So we did not find any other difference with any other biologics or any other comparisons. So what does this mean? So based on our findings, in general, there are no serious infections or less, but there are differences in serious infection risk among some of the biologics. And also our studies, our findings suggest that newer biologics are safer than the older ones, which need to be confirmed in further studies. So one thing we need to be cautious upon is that in our research we included only clinical trials. So as you know, some of you are already involved in clinical trials. So it is pretty strict, and so it may not be always reflective of real world. So because of this reason, we want to study this problem in the real world using real world people. So that's why we are aiming to do a study using the BADB data. So you have already heard about BADB. That's a good source of, a good resource for this particular research. I'm not going to talk about BADB, but point out few reasons why this is ideal for my research. It includes all the biologics that we currently use in the UK. It has pretty more number of participants in it. So actually more than 22,000 today. So from across the UK and Ireland. And it has a long follow-up duration. It follows up the participants for up to 8 years. That gives more information. And also, it includes serious infection data. So a serious infection is one of the primary focuses of this database. And so we have a comprehensive set of data to analyze and find the answer for this research question. So I'm already halfway through my PhD, so this is my next immediate task, and God willing, I'll be able to kick off with this study straight away, and hopefully I'll be able to present the findings in the next meeting, so with few other findings. So I take this moment to thank all my supervisors, all the co-authors who supported me, and especially once again I thank Psoriasis Association for funding this project and supporting me. So thank you all for coming and listening to this talk. Thank you so much.
Thank you very much. I think we've got time for one question. If there is a burning issue, there's one just over there. Thank you.
Thank you. Thank you very much for your talk. Julian Nash, Bristol Myers Squibb. Did you find any correlation with the duration of action or the half-life of the drug and the risk of infection? I was conscious of the fact that ustekinumab was the outlier.
So thank you for that question. We haven't actually looked at that particular aspect in this piece of study because we did a systematic review and we looked at the published literature. I think that's quite interesting to look at in our next study that we are going to look at using the BADB data. But with the published literature, we haven't looked into that. Thank you for that information, and that's quite interesting to know, actually. Thank you.
Thank you very much. Somebody asked me the other day, how did the association, how confident was the association that the investment we made in research was of practical benefit? And I think these two projects illustrate very well that it is not heavily buried laboratory science for the sake of it. This is practical research with practical results and hopefully practical benefits for future patient care. Thank you both for coming and presenting to us, and we wish you well for the rest of your studies.